Let's start with today's featured paper. Semaglutide-induced loss of skeletal muscle mass is blunted by co-administration of ketone esters. By Yasser Abutab and Jason R. B. Dyke from Cardiovascular Research Center, University of Alberta, Edmonton, Canada. This paper was selected because it addresses a critical and timely clinical challenge associated with the widespread use of GLP-1 receptor agonists for obesity, offering a novel and potentially actionable strategy to mitigate skeletal muscle loss, a significant adverse effect with long-term health implications. Here's a full breakdown. Semaglutide, a popular GLP-1 receptor agonist, has shown great promise in treating obesity. However, a significant drawback is that a substantial portion of the weight lost can be valuable skeletal muscle. This muscle loss is a major concern, as it can impact long-term health and mobility. Our research aimed to investigate if we could counteract this muscle loss using ketone ester supplementation and to understand the biological mechanisms involved. To address this, we designed a preclinical study using obese, glucose-intolerant mice. These mice were divided into groups receiving vehicle semaglutide alone, or semaglutide combined with a beta-hydroxybutyrate-generating ketone ester. Over 3 weeks, we meticulously assessed body composition, muscle strength, and endurance. We also delved into the molecular level, examining mitochondrial and atrophy-related gene expression in skeletal muscle samples to uncover the underlying mechanisms. Our findings were quite striking. Semaglutide monotherapy led to a significant reduction in lean mass and impaired muscle strength in the mice. At the molecular level, we observed suppressed mitochondrial gene expression and elevated atrophy-related genes. However, when ketone esters were co-administered, we saw a remarkable preservation of skeletal muscle mass and function. Importantly, this benefit came without compromising the desired fat loss. Mechanistically, the ketone esters prevented the adverse changes in mitochondrial and atrophy-related gene expression caused by semaglutide. In conclusion, our study provides compelling preclinical evidence that oral ketone ester supplementation can effectively counteract semaglutide-induced muscle loss, preserving both muscle mass and function. This protective effect appears to be mediated by preventing detrimental changes in mitochondrial and atrophy-related gene expression. These exciting findings suggest a promising strategy to improve the safety and efficacy of GLP-1 RA therapies. The next crucial step is to translate these results into human clinical trials to confirm their potential for patients. That was our featured study. Today, PubMed indexed 656 new open-access papers across 154 journals. We've selected 10 trending research areas and 10 studies for today's video. Let's look at the trending topics before we continue. Today's research landscape is rich with clinical innovations and advanced tech, but we're also seeing a surprising focus on research integrity and AI applications. Now let's see which papers stood out. Get this, ketone esters might actually blunt muscle loss from popular GLP-1 weight loss medications. And generative AI is evolving beyond language, becoming a general computing model for all of medicine. Now let's explore each of today's trends in detail, starting with number 1. Trend 1 of 10, clinical diagnosis and patient management. This trend focuses on improving diagnostic accuracy, understanding disease characteristics, and optimizing patient management strategies across various clinical conditions. This cluster covers 31 papers. Representative work includes risk factors for fecal incontinence after surgery for cryptoglandular anal fistula, protocol of a meta-analytic study, bacterial profile and antibiotic resistance in pediatric appendicitis, retrospective analysis and associated laboratory and clinical findings, and laryngeal leiomyosarcoma, a rare case report and literature review. Trend 2 of 10, molecular biology and biodetection. This trend applies molecular biology techniques to detect, characterize, and understand biological processes, agents, and their functions. This cluster covers 128 papers. Representative work includes validation of a pregnancy-associated glycoprotein-based lateral flow assay for early pregnancy detection in goats, molecular typing characteristics, and drug resistance of salmonella from foodborne diarrhea patients in Chifeng, China, and novel endopeptidase overcoming lysostaphin resistance. Trend 3 of 10, brain, cognition, and mental health. This trend explores diverse aspects of brain function, cognitive processes, and mental health conditions across different populations and influencing factors. This cluster covers 55 papers. Representative work includes stronger memories through smarter stimulation, bidirectional fractional order dynamics of the microbiota gut-brain axis in autism spectrum disorder featuring memory effects and inflammation thresholds, and financial precarity and mental health among French health students, a nationwide cross-sectional study. Trend 4 of 10, cancer biology and therapeutics. Research exploring diverse aspects of cancer, from molecular mechanisms and prognosis to digital health and diagnostic technologies. This cluster covers 55 papers. Representative work includes influence of digital literacy on digital health usage and tension among patients with cancer and healthcare providers, inter-observer variability across whole-slide imaging systems, and constructing a prognostic survival model for patients with stage 2 to 4 epithelial ovarian cancer, a study based on the SEER database and external validation in China. Trend 5 of 10, global health and social determinants. Examines how social, economic, and policy factors influence global health outcomes, particularly in vulnerable populations and specific disease contexts. This cluster covers 88 papers. Representative work includes effects of compulsory and free basic education policies and COVID-19 related educational disruptions on offspring mortality in low-income and middle-income countries. A quasi-experimental and modeling study. Understanding sugar-sweetened beverage tax implementation globally. A 34-year population-based observational study in 183 countries. And mais eats vous certain. Trend 6 of 10, therapeutic interventions for pain. This trend explores diverse therapeutic strategies, from interventional procedures to digital health, for managing various pain conditions. This cluster covers 53 papers. Representative work includes sympathetic and parasympathetic ganglion blocks for treatment of long COVID-19 symptoms, a scoping review, virtual reality for pain management during repeated pediatric laser procedures, protocol for a pilot randomized clinical trial, and ganglion impar neurolysis enhances the short-term efficacy of pudendal nerve pulsed radiofrequency treatment for pudendal neuralgia, a retrospective cohort study. Trend 7 of 10, clinical physiology and disease management. This trend explores clinical physiology, diagnostic methods, and management strategies for diverse diseases and their associated conditions. This cluster covers 75 papers. Representative work includes syndrome de Raynaud d'origine médicamenteuse suivant l'émorce d'un traitement par le succinate de metoprolol, l'hypotension orthostatique chez les personnes atteintes de la maladie de Parkinson, and longitudinal trajectories and predictors of glycated hemoglobin among type 2 diabetes patients concomitant with hypertension, spline-based linear mixed effects model. Trend 8 of 10, advanced materials and sensor technologies. This trend explores the development and application of novel materials and sophisticated sensing technologies for diverse detection and visualization purposes. This cluster covers 72 papers. Representative work includes automated synthesis of INSB quantum dots with improved batch-to-batch reproducibility via kinetically matched co-reduction, environmental detection of amoxicillin using a molecularly imprinted graphene oxide polyethylenamine electrochemical sensor, and dynamical analysis and exact solitary wave solutions of formula. See text. Dimensional integro-differential Jolent-Meillet equation using two analytical schemes. Trend 9 of 10, AI, optimization, and signal processing. Applying AI, optimization, and signal processing techniques to analyze, enhance, and develop systems across diverse domains. This cluster covers 72 papers. Representative work includes a generalization study in deep learning-based segmentation of lower limb muscles across different populations, a distribution-level statistical framework for reliable pipeline leak detection using multi-domain signal analysis, and enhanced optimization of photovoltaic units EVCSs and BESS integration in radial distribution networks using a hybrid sine-cosine gorilla search algorithm. Trend 10 of 10, post-publication corrections and retractions. Papers in this trend are formal notices of errors, concerns, or withdrawals of previously published scientific articles. This cluster covers 27 papers. Representative work includes in IRADA, retraction, identification of potent EGFR inhibitors from TCM database at Taiwan, and retraction, two birds with one stone, possible dual targeting H1N1 inhibitors from traditional Chinese medicine. That's the full research landscape for today. Remember, our number 1 paper, semaglutide-induced loss of skeletal muscle mass was already covered at the top. Now let's continue with papers 2 through 10, one by one. Up next, our second paper out of 10, Beyond Language. Generative Artificial Intelligence as a General Computing Model for Medicine by Arkadiusz Sitek and David W. Bates from Radiology Department, Massachusetts General Hospital, Boston, Massachusetts, USA, Harvard Medical School, Boston, Massachusetts, USA. This paper was selected because it proposes a groundbreaking conceptual framework for generative artificial intelligence in medicine, moving beyond language models to direct tokenization of diverse medical data, which has the potential to fundamentally transform personalized care and accelerate biomedical discovery. Let's break down its key contributions. Welcome. Today, we're exploring a groundbreaking concept in medical AI. This paper introduces a new way to handle complex patient data, moving beyond traditional methods. It proposes using direct tokenization of medical records, similar to how language models process words. This innovative approach aims to harness the power of transformer-based models to better understand and predict health outcomes. The authors outlined a conceptual framework for this new approach. They propose direct tokenization of medical data, breaking it into discrete units like lab results and vital signs. This allows transformer-based models to learn from the temporal structure of patient health timelines without relying on textual translation. An example model, Ethos, was highlighted for its potential in forecasting health outcomes and supporting clinical decisions. They also emphasized a privacy-preserving model-sharing framework to enable collaborative development across institutions. Up next, our third paper out of 10, Pharmacological Inhibition of LIF Signaling Reverses PD-L1, Mediated Immune Suppression in Gastric Cancer by Cristina Di Giorgio and Stefano Fiorucci from University of Perugia, Department of Medicine and Surgery, Perugia, Italy. This paper was selected because it identifies a novel pharmacological target, leukemia inhibitory factor, LIF, whose inhibition can reverse PD-L1, mediated immune suppression in gastric cancer, offering a critical strategy to overcome resistance to immune checkpoint inhibitors. Let's break down its key contributions. Gastric cancer is a significant clinical challenge, often showing limited response to current immune checkpoint inhibitors. This is largely due to the cancer's ability to evade the immune system. We investigated the role of leukemia inhibitory factor, or LIF, a cytokine known to alter the tumor microenvironment, in this immune evasion. Specifically, we wanted to understand its contribution to PD-L1 mediated immune suppression and if targeting it could be beneficial. Our study utilized both resected gastric cancer specimens and gastric cancer cell lines to investigate the underlying mechanisms. We employed various techniques, including immunohistochemistry and gene expression analysis, to quantify LIF and PD-L1. We also used TSNE analysis to profile specific cell subsets and conducted functional assays to assess T-cell activation after manipulating LIF signaling. Up next, our fourth paper out of 10, Supercharged ferritin nanocages enable universal cytosolic protein delivery by Dinkong Liu and Jun from Jiangsu Key Laboratory of Drugability of Biopharmaceuticals and State Key Laboratory of Natural Medicines, School of Life Science and Technology, China Pharmaceutical University, Nanjing, China. This paper was selected because it introduces a novel and highly efficient platform for universal cytosolic protein delivery, addressing a critical bottleneck in protein-based biotechnologies and therapeutics with broad implications across biomedical research. Let's break down its key contributions. Good morning, everyone. Today, we're diving into a critical challenge in biomedicine, getting therapeutic proteins inside cells. This is incredibly important for developing new treatments and biotechnologies, but current methods face many hurdles. Proteins often struggle to enter cells, bind nonspecifically, or get trapped in cellular compartments, limiting their effectiveness. This paper explores a novel solution to these persistent problems. The researchers developed a groundbreaking approach using supercharged interface engineering to create novel protein nanocages, specifically PFN+. These nanocages were designed to form non-covalent complexes with various protein cargos. They then tested this system in both cellular models and female mouse models to assess its ability to deliver functional proteins, including critical gene editing tools and therapeutic antibodies, directly into the cytosol. Up next, our fifth paper out of 10, Single-Cell Spatial Transcriptomics of Formalin-Fixed, Paraffin-Embedded Biopsies reveals colitis-associated cell networks by Elvira Manillo and Alexis J. Combs from Division of Gastroenterology and Hepatology, Department of Medicine, Mayo Clinic, Rochester, United States of America. This paper was selected because it introduces a robust framework for single-cell spatial transcriptomics on formalin-fixed, paraffin-embedded, FFP, biopsies, a significant methodological advancement that unlocks the comprehensive analysis of vast archived clinical specimens with preserved spatial context, thereby accelerating discoveries across numerous disease areas. Let's break down its key contributions. Ulcerative colitis, or UC, is a complex inflammatory bowel disease where understanding the precise location and interactions of cells is crucial. Historically, studying these intricate cellular networks in archived tissue samples has been challenging. This paper introduces an exciting new method, imaging-based single-cell spatial transcriptomics, or ISCSD, specifically adapted for formalin-fixed, paraffin-embedded, or FFP, biopsies. This innovation promises to unlock a wealth of information from existing clinical archives, providing unprecedented insights into disease mechanisms. The researchers developed a robust framework for applying ISCSD to clinical FFP mucosal biopsies. They analyzed samples from patients with ulcerative colitis, immune checkpoint inhibitor-induced colitis, and healthy controls. Using custom gene panels, this technique allowed for the precise detection of various cell subsets and disease-specific genes, while preserving their crucial spatial context. This innovative approach enabled them to map distinct cellular neighborhoods and transcriptional signatures associated with treatment response. Up next, our sixth paper out of 10, a deep learning system for bacterial identification and resistance prediction from MALDI-TOF data by Qiheng Wang and Qianchang Li from Department of Emergency Medicine, National Taiwan University Hospital, Taipei, Taiwan. This paper was selected because it introduces a novel deep learning system that significantly enhances the utility of existing MALDI-TOF technology by enabling rapid and accurate prediction of antimicrobial resistance, directly addressing a critical global health crisis. Let's break down its key contributions. Today, we're diving into a fascinating study that tackles the critical challenge of antimicrobial resistance. While MALDI-TOF MS is a powerful tool for identifying bacteria, much of its data remains untapped. This research explores how we can unlock that potential to not only identify bacteria faster, but also predict their resistance patterns, which is vital for guiding effective patient treatment. The researchers developed a novel deep learning system, named Antibiotic, to analyze MALDI-TOF MS data. They trained this system on a massive dataset, including over 89,000 records from both public and local hospital sources. The system employed different machine learning models, like light GBM and temporal convolutional networks, to specifically tackle bacterial identification and AMR prediction. Up next, our seventh paper out of 10, Global Health Ethics in International Collaborative Research During Policy Transition. What can we learn from the proposed newborn hepatitis B vaccine trial in Guinea-Bissau? By Kemanthri Moodley and Brian D. Earp from Department of Medicine, Faculty of Medicine Health Sciences, Stellenbosch University, Cape Town, South Africa. This paper was selected because it provides a critical and timely analysis of global health ethics in international collaborative research, addressing the foundational and continuously pressing problem of preventing exploitation and ensuring equitable partnerships in vulnerable populations. Let's break down its key contributions. International collaborative research, especially in Africa, has unfortunately been associated with significant ethical challenges for decades. This paper delves into a specific case, a proposed newborn hepatitis B vaccine trial in Guinea-Bissau. The core question explored is how we can ethically justify research that compares existing local care with a new global standard, particularly when health policies are in flux. This is a crucial area, as such research must navigate the delicate balance of scientific advancement and protecting vulnerable populations from exploitation. The paper analyzed the design of a proposed newborn hepatitis B vaccine trial in Guinea-Bissau. This trial aimed to compare the WHO-recommended birth dose vaccine against the existing local standard of care for newborns. It was planned to occur within a specific window before Guinea-Bissau's national rollout of universal birth dose vaccination. While the specific primary outcome wasn't detailed, the overall goal was clearly related to hepatitis B prevention. This design formed the basis for the paper's ethical scrutiny. Up next, our eighth paper out of 10. Magnetically guided biohybrid microrobots with multimodal locomotion for barrier penetration and multifunctional tumor treatment by Iji Gong and Imogen Saranyasa from Thrust of Robotics and Autonomous Systems, Systems Hub, the Hong Kong University of Science and Technology, Guangzhou, China. This paper was selected because it introduces a highly novel biohybrid microrobot platform with multimodal locomotion and barrier penetration capabilities, offering a transformative approach to targeted tumor treatment and drug delivery. Let's break down its key contributions. Microrobots hold great promise for targeted therapies, but a major challenge is their ability to navigate the body's complex biological barriers. Traditional synthetic microrobots often struggle with this due to their rigid structure. This research aimed to address these limitations by developing a novel approach to microrobot design, focusing on overcoming these physical and biological hurdles. Researchers developed innovative biohybrid microrobots by combining Euglena gracilis with magnetic components. This design allowed for controlled movement and deformation, while also harnessing the natural therapeutic properties of the microalgae. They tested these microrobots in complex environments, including 3D biological matrices and tumor spheroids, to assess their navigation and targeting capabilities. Up next, our 9th paper out of 10. VSIG4 suppresses anti-tumor T-cell immunity via the SLC3A2 dependent metabolic-ionic checkpoint by Guoling Huang and Chenhui Wang from the Key Laboratory for Human Disease Gene Study of Sichuan Province and the Department of Laboratory Medicine, Sichuan Provincial People's Hospital, School of Medicine, University of Electronic Science and Technology of China, Chengdu, China. This paper was selected because it identifies a novel immune checkpoint, VSIG4, and elucidates its mechanism of suppressing anti-tumor T-cell immunity through a SLC3A2 dependent metabolic-ionic checkpoint, thereby offering a critical new target for overcoming resistance to current immunotherapies in cold tumors. Let's break down its key contributions. Immune checkpoint therapies have revolutionized cancer treatment, but their success is limited in certain cold tumors, like pancreatic cancer. This highlights a critical need for new approaches. Our study investigated VSIG4, a protein implicated in tumor growth, to understand its role in suppressing the immune system and whether targeting it could improve anti-tumor responses. To investigate VSIG4's role, we utilized syngeneic tumor models and human VSIG4 knock-in mice, testing neutralizing antibodies against VSIG4. We measured tumor growth and survival and meticulously analyzed immune cell function using flow cytometry. Crucially, we delved into the molecular mechanisms, examining how VSIG4 interacts with SLC3A2 to affect T-cell metabolism and activation. Up next, our 10th paper out of 10, transdifferentiation of endothelial progenitor cells into rhabdomyosarcoma, defined by hedgehog signaling competence by Catherine E. Gadek and Mark E. Hatley. This paper was selected because it reveals a novel mechanism of cancer development through the transdifferentiation of endothelial progenitor cells into rhabdomyosarcoma, critically implicating hedgehog signaling competence as a defining factor. Let's break down its key contributions. Rhabdomyosarcoma is a challenging pediatric cancer, and its origins are not always clear. This study delves into a fascinating possibility that non-muscle cells, specifically endothelial progenitor cells, might transform into these aggressive tumor cells. Understanding this process and the signaling pathways involved could open new doors for treatment. To explore this, researchers likely used advanced cell culture techniques, manipulating endothelial progenitor cells to observe their transformation. They probably employed gene expression analysis and specific modulators to investigate the role of the hedgehog signaling pathway. The goal was to pinpoint the molecular mechanisms driving this cellular identity change. That's all for today's PubMed Daily. If you found this helpful, please like, leave a comment, and subscribe for daily paper summaries. See you tomorrow.